So, hello everyone. This is the first lecture of embryology, and in this lecture, I will be discussing the first week, which is the fertilization and implantation. Well, usually this is not the first lecture that other educators discuss, but I like to discuss fertilization and implantation and the further weeks because later, when you study the fertilization, the gametogenesis, a lot of things comes there, which is very easy to understand if you have understood, the development first few weeks already. So what I will be doing is first I will teach you implantation the first week, second week, all those successive weeks. Then I will go to the gametogenesis and fertilization. Well, trust me, I have tried this way and it works miraculously. So let's begin. First week. First of all, what happens in the first week? You all know there is coitus. Then there, all both the gametes, the secondary oocytes from the ovary will be released and the spermatozoa will be released from the semen. They will come, they will meet, they will fuse, and they will form the zygote. So this is fertilization. So the first thing which happens in the first week is fertilization. Once the fertilization has been completed, the product of fertilization, which is the zygote. Zygote will undergo successive number of division known as the cleavage and it will enter the uterus for the implantation. So the first two things which happens in the first week is what? Fertilization and implantation. Let's see. This is the uterus. This is the fundus. This is the body. This is the fallopian tube, right? In the fallopian tube, this is your infundibulum. This is isthmus and this is the ampulla. The junction that you see here, this junction is known as ampulo-isthmic junction, and this is the site where the fertilization takes place. I'm just giving you a brief overview. With that, we will start the first week, fertilization and implantation. So this is secondary oocyte, right? It came from the ovary to the interstitium. It was picked up by these finger-like projections known as the fimbriae. Then it went through the infundibulum, ampulla, and it reached this place known as the ampulo-isthmic junction. Similarly, spermatozoa deposited in the vagina. It moved through here. It went to the same place, ampulo-isthmic junction. Here, they underwent fertilization, and as a result of fertilization, they formed what? They formed zygote. Got it? Now, once this zygote has been, formed, what does a zygote needs to do? A zygote needs to be implanted. So in order to undergo implantation, it needs to go inside the uterine cavity or the uterus. But tell me one thing, why does it need to go in the uterus and why doesn't it implants itself in the fallopian tube? The thing is, the blood supply. The developing embryo will need, a plenty amount of blood, the nutrients and everything, which a uterus has the source. Uterus is highly vascularized from a large number of arteries, but not so good for the fallopian tube. So it needs to go to the uterus or the uterine cavity. One more thing which prevents it is that you all know one layer that is known as zona pellucida. Zona pellucida is a layer of transparent layer around the secondary oocyte and after fertilization around the zygote which prevents implantation. And this is the reason why zygote enters the uterine cavity, then it implants, because then the zona pellucida is removed. But if there was no zona pellucida, this zygote would have implanted itself in the fallopian tube. And this is a result of what? This is a case of what? This is a case of ectopic pregnancy. Got it? Great. So let's begin. Zygote. Well, zygote, usually people consider that this is a single cell, but this is not a single cell. What happens is, let's say this was secondary oocyte. and this is the spermatozoa, right? Secondary oocyte and spermatozoa. Spermatozoa will go inside. Now you see they have their both nuclei, right? Now you see these both nuclei will convert into pronucleus. I will teach you in the fertilization. But before these pronucleus fuses, the cytoplasm will start to divide. Now tell me one thing, is this a characteristic of a good cell that it started the division of cytoplasm even before the nucleus fusion has completed? No, right? Like here you can see the nucleus wasn't fused, but the cytoplasm has already started. So this is a reason why this is, this is known as a cell in disguise or intermediate cell. Thus I say zygote is a misnomer. I'm just clearing some basic concepts. We will go to the implantation just after 5 or 10 minutes. One more thing I told you, like this was the secondary oocyte. During the gametogenesis, I will teach you some cells over the secondary oocytes produce this layer of glycoprotein known as zona pellucida. Zona means zone, pellucida means clear. So this is a clear zone made up of glycoprotein secreted by some kind of cell during the development. This is secondary oocyte. But when the spermatozoa enters inside, and this is the zygote, the zona pellucida still persists over the secondary oocyte. Now this zona pellucida has a few roles. The first thing is it prevents polyspermy. Once a single spermatozoa enters inside the secondary oocyte, the zona pellucida undergoes certain changes which prevents other spermatozoa to enter inside. So one spermatozoa has already entered. This causes certain changes in the zona pellucida which prevents other spermatozoa to enter inside. So one function of the zona pellucida is preventing polyspermy. Another thing prevents implantation. One thing about the zona pellucida is that this is a layer of glycoprotein and it serves as a barrier. Let's say this is uterine endometrium and this is secondary oocyte, while this is a layer of glycoprotein. This is preventing this is preventing the secondary oocyte to communicate with endometrium and endometrium with the secondary oocyte. So if there is no, if there is no communication, how can endometrium say you come and you just settle down here? Endometrium doesn't say it to, spermatozoa. Why? Because it recognizes all the cells. Endometrium has the capability to recognize different cells. So it will only say it to the secondary oocyte, but it cannot say why? Because there is a barrier here. The barrier is glycoprotein. So this prevents implantation. Well, this is a very good thing as well as bad thing. The good thing is it prevents implantation in the fallopian tube. It prevents implantation in fallopian tube, thus it prevents ectopic pregnancy. You know what is ectopic pregnancy? Pregnancy outside uterine cavity. That is ectopic pregnancy. There was a case in which a female underwent, I think a CT scan or MRI, and it was found that a, entire developmental size, a mature fetus was found calcified in the pelvis of the female. So what happened was the child underwent ectopic pregnancy. It developed in the pelvis and it died. And after the death, you will study in the third year that the dead tissue undergo calcification. So the entire fetus was calcified. You can just Google it and you'll find it out. So there are two functions of zona pellucida: preventing polyspermy by certain changes once fertilization has taken place, and second is preventing implantation. Well, preventing implantation was a good thing to prevent ectopic pregnancy. But when this zona pellucida, when this, let's take this image, when this, just a second, when this zygote enters inside the uterine cavity, there are various factors here which causes the destruction of what? Destruction of the zona pellucida. So now since there is no zona pellucida, implantation can take place. So this process is known as the zonal hatching, which I will be teaching you later, right? Now, these are, I'm just establishing certain facts and basic concept. Now, there is something known as cleavage. What is cleavage? Cleavage means repetitive division of cells in zygote. So once the zygote has been formed, this will undergo 2 cell, 3 cell, 4 cell, 6 cell, and so on. This would be a repetitive fission division. Now you just, you saw, like, I didn't say 2 to 4, 4 to 8. I say 2 to 3. The thing is, these division are not the mitotic division, and this is unequal. Let's say there was 1, it became 2, 2 to 8 became 3, 3 to 8 became 5. So this is a fission reaction, but it isn't binary fission. The cell count doesn't increase as like 2, 4, 8, 16, and more. But among two cells, one is larger and divides, while the first causing it to 2 to 3 cell stays. Okay, two things completed. Now, once the cell has divided many times, like 2, 3, 4, 6, 8. So there comes a cellular stage 8 to 16. This stage is known as morula. Have you seen a morula? There are 8 to 16 cells arranged in form of cluster which looks like mulberry, and that is why this is known as morula. At 8 to 16 celled stage, the conceptus. Why do we say it conceptus? Because conception means pregnancy. So it is a result of it, result of fertilization. At 8 to 16 cell stage, the conceptus gives an appearance of mulberry. This is very densely packed and there is no cavity. Because later we will be seeing there will be a stage known as blastula. In that it has cavity known as blastocyst, thus known as blastula or the blastocyst. Now, still, this conceptus is still in the fallopian tube. This morula is still in the fallopian tube. At 32 celled stage, this is known as advanced morula. Advanced morula at 32 cell stage enters the uterine cavity. It still have zona pellucida over it, right? This is advanced morula enters inside the uterine cavity at day number 4, and just after this stage, this you know zona pellucida will be removed. So let's see. Late or advanced morula. It is 32 cell stage. Zona pellucida is still present. This is advanced morula because now the cells are arranged into layers. They are arranged into inner cell mass and outer cell mass. So we saw we had morula. Morula was just the cluster of cells. 8 to 16 cell stage. But here we have advanced morula at 32 cell stage, where the cells are arranged in outer cell layer or the inner cell layer, or the inner cell mass and the outer cell mass. And this advanced morula at 32 cell stage enters inside the uterine cavity at day number 4. Once it enters the uterine cavity, there are various factors which acts over the zona pellucida. Various factors communicate with the advanced morula, and it results in what? It results in the shedding of this zona pellucida. Now, once the zona pellucida is shed, what will happen? There will be uterine milk. This is not a milk, but this is a fluid in the uterus which is rich in nutrients, growth factors, and a lot of it. It will start entering inside because there is no zona pellucida to prevent anything. Zona pellucida was like a preventive barrier, but it's no more. So these fluid will start to go enter inside. Earlier we had outer cell mass and the inner cell mass, but what will happen is this fluid will push the cell at the periphery like this, forming a cavity. And once a cavity is formed, which is known as the blastocele, this stage is known as blastocyst or blastula. And this process of the removal of the zona pellucida, this is known as zonal hatching. And when does it take place? By the end of fourth day, because at the fourth day the advanced morula was entering inside. So it happens by the end of fourth day or the beginning of fifth day or even beginning of sixth day. Got it? So this was zonal hatching. We formed our blastocyst. How do we characterize a blastocyst? Blastocyst is characterized by the presence of a cavity, and the cavity is known as blastocele. Good. This is the stage that which doesn't have zona pellucida. So this will undergo implantation. Got it? So blastocyst has blastocele and it will undergo implantation. It will have structure like this. This will be the outer cell mass. This will be the inner cell mass. And here you will have the cavity or the blastocele. Got it? This was blastocele. Now let's go further and see our implantation in bit depth. Implantation. What is implantation? Suppose we have a plant, right? Now we have to put it inside the soil. This is known as implantation. Similarly, suppose this is uterine endometrium. This is blastocyst. We have to put it inside the endometrium. This process is known as what? Implantation, right? Implantation. At what stage implantation occurs? At the stage of blastocyst. At what day implantation takes place? It begins at day 6 and ends by day 10. So we saw in the fourth day, the advanced morula entered inside our uterine cavity. By the end of fourth day or the beginning of the sixth day, hatching of the, uh, our advanced morula took place, also known as the zonal hatching, by the end of fourth day to the beginning of sixth day. Now, implantation will take place from the sixth day to the 10th day. And the kind of implantation this is known as interstitial implantation. Another species have this partial implantation where the uh, implanted conceptus is not completely inside, but in human species this is completely inside the endometrium. That is why this is interstitial implantation. And where does the implantation takes place? Let's just take a photograph. In this photograph, what do you see? This is a child. This is umbilical cord going where? Here. This is the posterior surface. Here you have the fundus. So can we say the implantation takes place where? On the fundus, on the posterior wall. Always remember. What is the type of implantation? This is interstitial implantation. What is the site of implantation? Posterior wall at the junction of fundus and posterior wall. Like, if this is the sagittal section, if this is fundus, this is the body of posterior wall, implantation will take place somewhere here. Got it? This is the site of implantation. Now, once the implantation takes place, the outer cell mass will convert into what? Trophoblast. And the inner cell mass will convert into what? Embryoblast. Embryoblast, because this is going to form our embryo. So this was our first week, the fertilization and the implantation. Now we will begin with the second week, which is the formation of the bilaminar germ disc. See, till now, what we saw was, let's say, this was our outer cell mass, right? Now the outer cell mass after implantation, we were calling it what? Trophoblast. And we had our inner cell mass, which had converted into what? Embryoblast. Now, the embryo has rich vascular supply. It is receiving large amount of glucose, amino acids, and all those necessary biomolecules. So it will undergo profuse level of cell division and differentiation. The structure which you see here, the trophoblast and embryoblast, will undergo differentiation, right? And here you have the cavity. This cavity is what? Blastocyst. Now, this is the implanted structure. Now, this will form our bilaminar germ disc as it shifts to the second week. Again, trophoblast. Let's just draw it like this. This is our trophoblast. Got it? And here, we had various cells. The embryoblast. Here we had our cavity. These embryoblast cells, the red color cells that you see here, they will arrange in a flat manner like this. Or you can say a large number of embryoblasts will start lining the blastocyst. The rest number of cells will grow over these cells. These are columnar in nature. Now the cell which is growing upon these cells, these are known as epiblast, while the cell lining the blastocyst, this is known as hypoblast. Why didn't I name it earlier? The thing is, see, hypo means below, epi means upon. So the cell from the embryoblast which is lining the blastocyst, those are hypoblast, and the cells which grow upon the hypoblast, they are known as the epiblast. Got it? Soon what will happen is these epiblast cells will start to migrate and will start to line the inside surface of the trophoblast. And these migrated cells which are now lining the cavity, again there will be a new cavity here. These cells are known as what? Amniogenic cells. So they were the epiblast cells which migrated, lined the inner surface of the trophoblast, and these are known as the amniogenic cells. Like something sparkled, right? Amniogenic, amniotic. So yes, they are lining a cavity known as amniotic cavity. So what is lining the amniotic cavity? Amniogenic cells and epiblast. They are lining the amniotic cavity. Similarly, we had hypoblast cells, right? We had our hypoblast cells. Now these hypoblast cells will also migrate and will also line the trophoblast cells from inside. Do you know what is the name? The name of these migrated differentiated cells are known as Huesner cells. These were hypoblast, and the cavity which they are surrounding is known as, yes, yolk sac. So epiblast migrated, formed the covering from inside, enclosed the cavity, amniotic cavity. Hypoblast migrated, lined the trophoblast from inside, enclosed the cavity, yolk sac. Understood? Now, do you see one thing? We had made two layers here. One layer of epiblast, one layer of hypoblast. So one disk of epiblast here, one disk of hypoblast here. Can we say it is two disk or bi-layer disk? What is the other name of layer? Lamina. So can we say this is bilaminar disk? This is the reason why second week is known as the week of formation of bilaminar germ disk. This epiblast in future is going to give rise to what? And hypoblast, they are going to give rise to endoderm and ectoderm. Got it? Great. Now, you understood this much part? Now let's do one thing. What I want to do is, uh, I want, I already have notes. So let's try to understand everything on the notes, right? So a quick revision here. We had trophoblast, embryoblast. Some cells of the embryoblast started, differentiating, and they differentiated to form what? Hypoblast. While the rest also differentiated sometimes later and they formed what? They formed the epiblast. You see this layer? Soon the epiblast cells migrated towards the periphery. They formed this amniogenic cells. Soon the hypoblast cells migrated towards the periphery. They formed what? Huesner's membrane. They enclosed amniotic cavity and yolk sac. Got it? And this two-layer disc is known as bilaminar germ disc. Now see, we have this cavity known as the yolk sac. Some cells from the yolk sac will start migrating outside. Where will they migrate? Suppose this is the layer of trophoblast cells, this one, and this is your Huesner membrane. This cell will start the migration from the yolk sac and will migrate and get deposited between what? Epiblast and Huesner membrane. This will divide, differentiate, and this will give rise to your primitive mesoderm. And this primitive mesoderm is usually named as extra embryonic mesoderm. Why? Because this is outside the embryo. You remember embryoblast giving rise to embryo. So this has come outside of it. So this mesoderm will be known as extra embryonic mesoderm. You see, at this stage we have primitive ectoderm, we have primitive endoderm, and we have extra embryonic mesoderm. Later, we will study during the systemic embryology. Everything comes from ectoderm, mesoderm, and endoderm. So, is there any difference or it's just same here? See, there is a difference. There we will see three layers. There will be ectoderm, mesoderm, endoderm in a single disc. That would be trilaminar. But right now, you have one disc, another disc, and the third layer, the mesoderm, it is not present inside the embryo. This is present outside it. See, this is epiblast or the primitive ectoderm. This is hypoblast or the primitive endoderm. And the mesoderm is present outside. Got it? If this is clear, let's go further. Soon, in this extra embryonic mesoderm, small cavities will start to come. Small cavities will begin to come, right? Many of these cavity will start to fuse. You see, there are many cavity. Soon these cavity will start to fuse. This fused cavity is known as extra embryonic coelom. What is it? We had extra embryonic mesoderm. Small cavities came inside the extra embryonic mesoderm. Those cavities fused, and upon the fusion, they gave rise to a coelom. Coelom means cavity. Now, this cavity divided the entire extra embryonic mesoderm, you can see, into outermost part and the innermost part. You can see this cavity divided our mesoderm into outer and inner mesoderm. Is there any significance? Yes, we will study it. But for now, do you see at this part there is no division. Here the extra embryonic mesoderm is not divided into outer and inner part. In fact, there isn't even a cavity here or a coelom. Why? Because this is undifferentiated extra embryonic mesoderm, and this part is known as, what is it? Connecting stalk. In future, this is going to be the part which will give rise to what? Umbilical cord. So let's consider for right now. This is your light bulb. This is the wire which is giving it electricity. Where is it entering from? Here, the connecting stalk. Got it? This part. Now, here you have to understand a few things. These are terminologies. You will see yolk sac is going to give rise to a large number of your visceral organs. Visceral organ is also known as splanchno, right? So the extra embryonic mesoderm, which you see here, around the yolk sac, this mesoderm is known as splanchnopleutic extra embryonic mesoderm, right? While the mesoderm, which is present around your amniotic cavity, this is known as what? Somatopleuric extra embryonic mesoderm. Got it? Somatopleuric extraembryonic mesoderm, splanchnopleutic. We will study the, somatopleuric intraembryonic mesoderm also, splanchnopleuric intraembryonic mesoderm also. But this will be present between these two layers. Okay. For now, the part of extraembryonic mesoderm around the yolk sac is known as splanchnopleuric extraembryonic mesoderm, and the part of extraembryonic mesoderm around the amniotic cavity is known as somatopleuric extraembryonic mesoderm. Now, another thing, right now this might seem you like I'm teaching too much, but the thing is, bacha, this is completely based on Langman embryology. That is like the fundamentals of your embryology. AIMS Delhi or any prestigious medical university follows this book. So I think if you are going to become a doctor, become a good doctor. A good doctor always follows, the standard medical textbook, because those are the books which are the best. They are the best. They don't have, the errors, typos, and everything. Yeah, they can update, because they are, there are always changes, but they are always updated to the best possible information available to the medical sciences. Got it? So this might seem you extra, but trust me, this is the lecture one. This is not going to be, like the end. This is the beginning. You have come to learn here. You will start learning, and as the day passes, lecture one, lecture two, and so on, everything will get easier, because you are coming from your high school basically. So you have the level of embryology from your NCERT. NCERT is nothing really. They have discussed embryology in just one or two pages. It's nothing. There is an entire book here. So you can't expect to give you just little information besides NCERT. This is medical sciences. You are going to become a doctor. When you are seeing a child, you should you should be able to see, like, imagine, let's say it's heart. You, in your mind, it should run like how it came from the amniotic cluster, how the heart tube formed, the how the sinus bulbus, all those circulations formed, the folding of those circulations, how everything formed, how it's working, the structure, the histology, the physiology, and everything. If you get disappointed, like this is too much information, then it's not good for you, right? So आदत डाल लो आप, get used to it. You are going to become a doctor, and you have to read it. You might read it the easier way or the difficult way, but you have to read it. And I am trying my best to simplify everything with these beautiful diagrams, schemes, and everything. And this is just embryology. When I come to anatomy, I will show you everything, like the anatomic atlas images, the cadaveric images. I will draw beautiful schemes, everything. Just give me time, give me your attention, and I will do everything. I will literally write everything on your brain like a blackboard. Well, it's not black, so don't worry. Well, it's black because it's gray matter, right? So just give me your attention and I will do everything. Just make yourself available to it. Now you see here we have two different terminology. The two different terminology is amnion and chorion. A very important thing about the systemic embryology. What is chorion? Chorion is your trophoblast. Your outer cell mass had differentiated into trophoblast. So this trophoblast with the somatopluric extraembryonic mesoderm. Similarly, you have amnion. What is amnion? Amniogenic cells plus somatopluric extraembryonic mesoderm. I just told you this is your splanchnopleuric extraembryonic mesoderm, and this is your somatopluric extraembryonic mesoderm. This entire part that you see here. This is somatopluric and splanchnopleuric extraembryonic mesoderm. Now this somatopluric extraembryonic mesoderm, along with the layer of this trophoblast, this is known as chorion. While this amniogenic cells plus somatopluric extraembryonic mesoderm, this is known as amnion. I hope now you understood. What is amnion? Amniogenic cells plus somatopluric extraembryonic mesoderm. What is chorion? Trophoblast plus somatopluric extraembryonic mesoderm. In this photograph, it's very easily shown here. So with this, we have completed first week, which was mostly fertilization and implantation, and the second week, which was the formation of the bilaminar embryonic disc made up of epiblast and hypoblast. Got it? So what you have to do is just read the notes. Everything is already there. Once you read it, you will understand everything. And if you come up with any doubt, I'm really available to you. So just let me know if you come up with any doubt, just text me. I'll be there to help you. So have a good day. Sleep well, rest well, study well. Sleep is very important for you, and the same is your food. So please do well. Have a good day. Goodbye.